Hello everyone, welcome back to class. This will be the last video for research methods in pharmacology. So, as I left off saying, we are going to finish up with covering behavioral pharmacological research methods. In other words, they're pretty much usually ways that we use animal models to mimic human behaviors and pathologies. There are three big things we look for when saying an animal model is good. We look for construct validity, predictive validity, and face validity. You will need to know these three validities. What is each one? How does it compare? Okay. Construct validity. The similarity between the methods by which the model is induced compared to humans. Okay, for all of these, you can add compared to humans at the end, okay? So the genetic and environmental factors, are they similar or different than humans, okay? If they're similar to how the disease presents itself in humans, we would say it's good construct validity. Predictive validity. How good the model is at predicting the effects of drugs. So this is the one we'll be talking about a lot in this class because we like to talk about drugs here. So how good does the, say, antidepressant treat depression in animals versus humans, okay? If the effects are similar, it's good predictive validity. If the effects are different, that would be poor predictive validity, okay? This is, again, assuming that this effect is based on shared mechanisms, which, we wouldn't use it on an animal that doesn't have the same mechanism. So we just assume that. We make a lot of assumptions in science. Finally, we've got face validity. So this is how good the animal model is at recreating the symptoms of the disease, okay, or disorder. These could be anatomical, behavioral, neurochemical features of the disease, okay? So if we see decreased gray matter in humans, then we should also see decreased gray matter in animals, okay? That would give us good face validity. The same symptoms we see, or, the same withdrawal effects, those could still be face validities. Now, using animals in research does have its own challenges because there are some diseases and disorders that just don't exist in animal populations. So like a schizophrenic mouse, that's not really a thing, right? Or like AIDS even, so going outside of neuropsychopharmacology. HIV and AIDS is a human disease or a primate disease. So we can't use a mouse model for that, right? We do have these behavioral tests that have been designed to provide a measurable proxy for human behaviors so that we can look at very common behaviors that are common to multiple disorders and diseases and look at the face validity, predictive validity, and construct validity of these animal models. So first, we have different types of learning and memory mechanisms or tests. One of the most famous would be the Morris Water Maze spatial test. Again, you should have learned about this one before, but for those that need a refresher, there's a water maze down here. It has murky water in it so that the rat or animal can't see through the water. And then there's a platform somewhere hidden in the tub that if they swim around to, they can stand on and, not have to swim. Now, we use this for learning and memory to see if the animals will use their spatial awareness to find the platform easier on subsequent trials, because they should remember where the platform is. So decreased time to find the target platform in repeated trials indicates good spatial memory. We can also use mazes So this is the radial arm maze or the radial 8-arm maze, where there's a lot of different configurations we can do with this, but one of the main ones is, there'll be food on each arm, the rat's placed in the middle, they go, great, they'll be taken away for a bit, they'll go back in the middle, and what we should see is that they'll remember, well, last time I went here, so now I should avoid that arm because I know there's no food there. I've already eaten that food, I should go somewhere else. And that should show us that they have good memory. They remember where they've been before. Or if they're in this and the food's always placed in this arm every single time, then, the next day they should go here first because they know this arm always has food, that kind of thing. So we can do a bunch of different things with these radial arm mazes. And then finally, we've got the novel object recognition task. This is to evaluate episodic memory specifically. Pretty much the animal will be presented with 2 different objects here. They get to explore them, become familiar with them. It's great. Then on the next trial, one of these objects will be removed. And what we should expect to see is rats and mice are very curious. They like to explore new things. So they should spend more time with the new object rather than the old object. Okay, so if we put a new object here, so say it's a purple dinosaur, this mouse should spend more time with the purple dinosaur. And that would indicate that the animal has good memory because it remembers that this one's new. I don't need to explore this little red suitcase because I already know about that. Okay, so if the rat spends an equal amount of time at both things, that's indicating that rat does not have good memory. Up next, there's a bunch of tests that we can use to measure anxiety levels in animals. A very common one is the open field test. That's pretty much what this is here. You can see it's just a big open square, and then there's some lines that aren't there in real life usually. And we just let the rat explore. Most rats and mice, they will explore all over, but they tend to also have a fear of open spaces. So you'll see they tend to spend most time on the edges, but they will go out there, okay? They want to explore, but they're going to spend more time on the sides. And so we can track their movement usually with a camera. If we see decreased time in the open area and more time on the side, so we lose this exploration in the middle, that indicates an increase of anxiety. So a little bit of exploration in the middle is normal. Once we lose all that exploration in the middle, that indicates high anxiety. Up next, we've got the elevated plus maze. So you can kind of see here, it continues. So you've got 2 open arms and 2 enclosed arms. The rats will be placed in the middle. They'll be allowed to explore. They usually will explore all of them. Entry into these open arms indicates novelty-seeking behavior. So a non-anxious mouse, a non-depressed mouse will come out here. It wants to explore. An anxious or depressed mouse will not. They will spend more time in these enclosed ones because they feel safer. They don't feel as exposed. We can also induce fear in animals. One way is the fear conditioning paradigm. So here's just a little shock box. You'll see there's metal rods on the bottom. There's a chamber here, a chamber there with a little opening. There's also usually lights on both sides that can be turned on or off, or there'll be some sort of tone. So usually the animal's presented with a signal, like the light goes on, and then there'll be a foot shock. Okay, the animal should learn to fear the light or the tone that is paired with the foot shock. We'll see that when the light goes on, this rat will freeze. It won't try to avoid it or something because that's the fear response. So that one's very common. We can also use a similar thing, but only with one chamber for operant conditioning. So we can do reward operant conditioning. So if the animal does something we want, like press a lever, they get rewarded with food. Sometimes, especially in drug experiments, they'll actually have little tubes attached to their brain, and when they do the correct behavior, they get rewarded with the drug. So rats can do a behavior and get rewarded with cocaine, and they become addicted to cocaine, so they'll do more of the behavior to get more cocaine. It's a whole thing. But we're using operant conditioning, so you should have learned about this, again, an introduction to it in 104, but more about it in Psych, was it 282 and 381, all cover operant conditioning and learning. So you should know all about this kind of Skinner box idea. And then finally, the very last of the behavioral pharmacological research methods is using conditioned place preference. So this is a Pavlovian conditioning paradigm. So this is classical conditioning. So the other one was operant conditioning. This is classical conditioning. So here we have a drug injection is paired with a neutral environmental stimulus to create a conditioned response. So sometimes it's just the area that they are, and sometimes it's a tone or something like that. So in this paradigm, then we've got a 2-chamber box, or sometimes there'll be a 3rd chamber over here, and then there's a 3-phase protocol. So we've got preconditioning or habituation, so the animal is allowed to explore all of the chambers as much as it wants for 15 minutes over 3 days, and then conditioning. So alternating injections of drug and vehicle in altered chambers. So say here, they'll get an injection of the drug. When they're in this chamber, they'll get the vehicle. Remember, vehicle is just whatever the drug is dissolved in. So if the drug's dissolved in water, the vehicle is water. Usually the vehicle is some sort of cerebral spinal fluid, so we might call it artificial cerebral spinal fluid. We might call it saline solution. Saline solution is an artificial cerebral spinal fluid. It's also just a salt water, pretty much. That's what a vehicle is, okay? And it's used really as like a control. So vehicle gets injected, drug gets injected, vehicle gets injected, drug gets injected, that kind of thing. We then do a post-conditioning test, so open exploration, and we see how much time does the animal spend in each chamber. In general, we tend to see when it's a drug that they like, so usually like cocaine, then they'll spend more time in the chamber where they receive the drug because they got that good thing. So even though nothing bad happened here, they still prefer over here, okay? So that's the conditioned place preference. Again, this is one that we'll see a lot in this class because it's great with drugs. Now, I did include some unit summary slides about pharmacokinetics, pharmacodynamics, and the research methods. So you can read through those, similarity and differences, the key terms between the things, and tolerance, of course. So that is it for today's lecture. We will be starting with our talk on different drugs on Friday's class. That should be January the 14th.